Thanks very much for the invitation. Great to join this seminar. Hopefully this sharing is now working. Okay, let me know if you don't see the slides. Okay, so I'm happy to discuss some of the work that we've been doing in the lab with kind of sequence to function or sequence to activity models. And for this audience, I don't think I need to explain any background on these kinds of models. But I'll say the perspective that our group and kind of from my background, the perspective that we have on these models and what we're trying to do with them is to use them for kind of clinical genomics motivations and personalized medicine motivations. And for that, you want to be able to apply these kinds of models to genome data from individual people. And so this is kind of the personal genome interpretation problem that we're interested in. And if you have a patient coming to a clinic and they get their genome sequenced, let's say they have a rare disease or any other phenotype that you want to predict, can we use these kind of models, plug in that person's individual mutation data and kind of understand what's going on molecularly inside their cell using the kind of sequence input, but with the personalized genetic variant information added in. And these models should be able to predict things like gene expression, chromatin states, et cetera, from that person's sequence information. So when we started working with these kinds of models and trying to use them for that type of problem, we realized that a lot of the evaluations that had been done on these kind of models up to that point had been looking at their performance across different regions in the genome. So the kind of cartoon version of the way the models typically were evaluated and also how they're trained is that you have kind of different regions along a genome. So this is just a cartoon of two different genes. Let's say it's a gene expression, a model that predicts gene expression. You have kind of different genes along the genome, and the expression prediction or whatever molecular phenotype you're predicting with the model, that prediction is getting made based on the sequence surrounding that gene or that region of the genome. And so these are going to be kind of relatively different sequences. They obviously have some shared elements, but different sequences at different places in the genome. And so the training is done across these different chunks of the genome. And then most evaluations had also been holding out regions of the genome and evaluating performance in those held out regions. So for applying these models to understand personal genome variation, it's a bit different of a problem in that what we want the models to be able to do, and we want to evaluate, if they work well for this task, is to look at a given gene. So let's say this gene on the left, we have kind of that gene with the expression level and the genetic information from a given person, and then we'll have data from another person, which is almost the same sequence, but just with some small changes. So this is same gene, but just a small amount of genetic variation between different people. And they might have different expression levels that's regulated by that genetic variation. And we want to apply the models in this case to predicting differences between individuals. So it's kind of a separate axis of variation that we want to explain for this kind of clinical or personalized genomics applications. And so it wasn't clear at the time if the existing models, the way they had been trained and evaluated, if they would work well for this kind of alternate axis of variation. In theory, they should, if they've learned to understand the regulatory grammar of the sequence that controls gene expression, you should be able to plug in kind of any variation that you find in a new person and get a good prediction. So it's reasonable to expect that they could handle this problem, but the scale of the variation is a lot different. There's only a small number of differences between people rather than there being kind of overall different patterns of sequence between two different genes in the genome. And so it's a bit of a harder problem in the sense that the models have to be more sensitive to this small amount of variation. So that's what we set out to evaluate initially. And so we just set up an evaluation where we had some paired personal genome data and transcriptome data. And we would, for each person, just plug in kind of all of the genetic variation within the input context window of the model that we were evaluating. And we evaluated several different models that existed at the time that were trained to predict expression as one of their outputs. And we had the kind of paired transcriptome data from RNA sequencing for each of these individuals, for each of these genes. And so we end up, with setting up this evaluation, we end up with a matrix of individuals by genes of all of their predicted expression levels from these models that we're evaluating. And then we also have all of the another matrix in the back here of all the measured expression values for those individuals and by genes. And then we can compute this correlation. We can compute the model performance and how well it is able to predict expression in kind of along these two different axes. We can compute the correlation for a given individual across all the different genes that are predicted for that person. And that's kind of equivalent to how the models had been evaluated previously. But we could also look at this orthogonal axis and compute the correlation for a given gene across individuals. Okay, so we did this initially just with this Ioannidis consortium data. So this is publicly available data from 1,000 genomes individuals, where the RNA sequencing is from the lymphoblastoid cell lines that were created from these people. And in order to be kind of fair to the models, we limited the set of genes we were looking at to genes that had a significant EQTL in the original analysis performed by that consortium. So we know there should be some genetic regulation of expression for that set of genes. And this work was led by these 3 undergrads in the lab initially. Okay, so first, just as a sanity check, the kind of correlation across genes for each individual. And for that, since we're computing it for every individual, we end up with kind of a histogram of all these correlations. And that looks like this on the right. This is for Informer, which was one of the 4 models that we tested that had the slightly better performance than the others that we tested at the time. And for Informer, you see there's a pretty tight distribution here of these values of correlation across genes. And the mean is around 0.55. So this is in the range of kind of what we expect from what had been known about these kinds of models, that they do explain a large amount of variation across different genes in the genome. Okay, but then when we looked at the correlation in this other direction across individuals for a given gene, now we also have kind of a histogram over all of the genes where we compute this metric. And here, so this is the distribution that we get for Informer. You can see 0 is here in this black line, and then the mean is very close to 0, slightly shifted to the right, so a little bit on average of positive correlation, but the mean is very low. And there's a very wide distribution across genes. If you look at kind of the x-axis here, there are some genes that have very strong correlation across individuals, but then some genes that have a very strong negative correlation. And so I'll get back to that in a minute. But basically, the mean was close to 0, which we were a little bit surprised about initially, that there seems to be not actually that much signal for this particular type of task if you set up the evaluation framing in this way. And so we wanted to investigate kind of what's going on here. What does it tell us about what has been learned by these type of models? And I should say this was general across all of the 4 models that we tested. So it's nothing specific to Informer or any one model. It seems to be a general property of models that are kind of trained in this way of setting up the training as different windows along the genome. So this is a plot of the 4 different models that we had tested at the time. On the left is those kind of mean and standard deviation of those correlations across genes. So they're all kind of around 0.5-ish. And then the correlation across individuals for each of those models, mean and standard deviation. So these are wide error bars. These are standard deviation, not standard error, but reflecting that very broad distribution of correlations that we saw for the different genes. And so you might ask, and what do we expect that correlation to be? Because correlation across individuals is a harder problem, and it's not all genetically regulated, right? There are environmental influences on expression that make it different between different people. So you wouldn't expect to necessarily explain all of that variation just from the genome sequence. But we can have a baseline of what we would expect using some variant-based models that have been used before for TWAS, for people that know, so transcriptome-wide association studies, where you just use kind of this linear combination of variant dosages near a gene. So yeah, these kind of models have been proposed to do these association studies with imputed gene expression in a GWAS cohort. And they are just set up and trained in the same kind of data that we did this evaluation on, where you train just this simple linear model where you have the dosage, how many copies of each SNP or each variant each person has, and then you train kind of a set of weights for each of the SNPs. And there's a few different proposed models that are just slight variations of this. Kind of the early one that was proposed is called PredictScan, and to fit the betas in that specific proposed implementation, they used elastic net regularization. And so we just trained our own kind of PredictScan models to use as a baseline to let us know kind of if you're seeing data from a particular gene and you're seeing the paired genetic variation and expression data, and you train just this simple linear model, what's the performance that you expect to be able to get from doing that? And so if we do that just as a baseline here, this is the purple bar where you do see, it's still a relatively low, it's 0.2 something for the correlation, but it's much higher than you get from these kind of deep learning models that were working at the sequence level rather than the variant level. And so we expect that if the sequence-based models really have learned the regulatory grammar in a way that's generalizable to this cross-individual task using the variation between individuals, they should at least be able to get kind of this level of performance that we see from these variant-based models. And we think of it kind of as a minimum that you should be able to achieve, because these are just linear combinations of SNPs, not including any interactions, which you should be able to model with the deep learning models. And also these are really only using common variants, because you can't train the weights, the betas, for variants that are rare in the kind of cohort that you have to train. So ideally, the sequence-based models, you're plugging in all of the variants for a given individual, whether common or rare. And many of the rare variants are expected to have large effect sizes on expression. And so you should be able to capture that as well. So you should be able to kind of beat this baseline, ideally, if you can kind of get the models to work in this setting. Okay, so what's going on here? And we try to dig in a little bit to see where some of the failure modes are. One kind of sanity check that we wanted to do is just, are we seeing kind of any real signal based on what we expect from the individual variants that we had seen in these studies, in the EQTL studies, to be associated with expression. And so we just stratified the performance. This again is for informer, and on the y-axis is the correlation. Each dot is a gene again. So for each gene, the correlation that we saw across individuals for the informer predictions. And if we stratify those genes by how strong of a signal they had in the original EQTL analysis, so on the right is a more significant EQTL that those genes had. And so if we stratify performance that way, we do see that there's this trend towards the genes that have a stronger EQTL signal having higher correlation with informers. So that's a good check that there is something being learned about these genetic signals. The genes that have a stronger signal kind of is getting picked up to some extent. But if we look, so this is just showing the genes that have the positive correlations across individuals. If we look at the negative correlations, it was interesting. We also saw kind of this similar trend that some of the genes that had very strong genetic associations from the EQTL analysis had very strong negative correlations predicted by the informer model. And so this is telling us that there is a trend toward having these higher magnitude correlations with stronger EQTLs. But it seems to be that the trend is, it's sometimes in positive, but sometimes in negative direction as well. So sometimes the models are picking up the fact that there is a regulatory variant that's present in these individuals, but they're getting the direction of effect of that variant on expression incorrect. And so we wanted to know, is there something about the regulatory grammar of those genes that have this like very strong negative correlation? Maybe there's something going on with the specific regulatory elements that those variants are in, or the specific way that the variant affects the regulatory element that's hard for the models, or that the models learn in this opposite direction for some reason. And it's like something particular to that type of regulatory grammar. And if that were true, then we would expect that there would be the set of genes that have these negative correlations should be similar between all the models. They should kind of get that incorrect direction for the same set of genes. So what we did was look at the performances of these cross-individual correlations for pairs of models and compare them to each other. So that's what I'm showing here for Informer compared to a couple other models, Basenji and Expecto. And so hopefully this kind of comes through in the plot. You can see, again, each dot is a gene, and we're looking at kind of how the performance from the two models compares. And so you would expect to have this like increased density along the y=x line here. But we also see a bit of an increased density along y=-x. So the data are almost forming this kind of X shape. So we tend to call them these X plots in the lab. And our interpretation of these X plots is that there's kind of more agreement between the models on the magnitude of this correlation across individuals, meaning on the kind of magnitude or the presence of regulatory variation around these genes. There's more agreement on that than on the direction of that variation's effect on the expression for a given gene. And so it seems like this hypothesis that maybe the regulatory grammar is such that models are all learning it as opposite, that seems to be not the case. And it's more often the case that there's just some inability to assign a direction correctly, and it's somewhat arbitrary which direction is getting assigned by a given model. And we repeated this also with a set of replicate models of Basenji that we just trained in-house with different random initializations of the model. Everything else the same, like same architecture and hyperparameters and same training data. And we just like retrained several versions of Basenji. And they also have these X plots if you look at their performance. So we think there is this something going on with being able to assign kind of what the direction of effect of a small genetic change is when you're looking at expression as an output. Okay, so we wanted to kind of dig into what's going on with that. And one thing, so this shows that it's not a specific regulatory grammar that's hard to learn, but it may be that there are certain types of grammars that are giving rise to this kind of more arbitrary assignment of the direction of effect. So it could be regulatory grammars that are harder to learn the direction in a way that makes it arbitrary rather than a way that makes it kind of always learned in the negative direction. And so we wanted to explore a bit kind of what's going on with that. And there had been work also from people on this call about how many of these models are not utilizing distal information very well. And so because we're predicting gene expression here, we wanted to see, is it more challenging to predict expression and kind of assign the direction of effect of variation in the input sequence? Is it harder to do that if the variation is further from the start site of the gene? And so we did this analysis also with that same set of Basenji model replicates that I mentioned that we had trained just with different random initializations. We did an experiment where we took a starting sequence and kind of either plugged in a TF motif and looked at the difference in performance, the difference in predicted expression, plugging in the TF motif versus the original sequence. And then we also did a kind of second evaluation where we plugged in the TF motif, and we also plugged in a mutated version, and we chose to mutate kind of the most conserved or the lowest entropy site within the TF motif. So hopefully putting in a variant that should change the impact of that motif significantly. And so we also looked at kind of the delta between the prediction on expression from this motif inserted sequence and the mutated motif inserted sequence. And this was done by 3 grad students in the lab. Okay, and when we do this, we kind of varied the insertion position. So how far upstream of the TSS we inserted this motif and the mutated motif. And what we're plotting here is how inconsistent the predicted direction of effect of that insertion, either in kind of the light gray, the inserted motif versus the background, or in the darker gray, the mutated motif versus the unmutated motif. And this is, as I mentioned, plotting kind of how much those replicate Basenji models disagreed with each other about the direction of the effect on expression from doing those insertions. And you can see that as you go, as the insertion gets further from the start site, you see this much higher rate of disagreement between the models. And you don't have to get kind of that far away. We went out to 10 kb, and you start to see kind of a lot of disagreement. So it's likely that, this issue with TSS distance, which, as I mentioned, has been seen, by other groups as well, that's contributing to this challenge of being able to predict expression across different individuals and understand the effect of mutations or variation between those individuals on the expression levels. So how do we want to, or how could we address this problem? So we tried a couple of things, and I'll get back to the TSS distance issue in a minute. But one clear thing that made sense that we should try, if you think about how these models are trained, is that they're trained on reference genome data. And with the reference genome and then lots of molecular data that's been obtained from a bunch of different cell lines, but they haven't seen variation during training. And so the question was whether if you retrain the models or fine-tune them and allow them to see some variation on the scale of the amount of variation that you normally see between personal genomes, if they get to see that kind of during training or fine-tuning, does that improve their performance on variation in kind of held out individuals or held out genes? So we set up a fine-tuning framework where we took the informer model, and then we used the same data set that I had already presented the results from, where we have the whole genome sequencing, so the personal genome data from all the individuals and the paired kind of RNA-seq transcriptome data from those individuals. And in that data set, I should mention that most of the populations that were represented were European populations, but there's also the Yoruban population as well. And so when we did this fine-tuning, we were able to do a few different splits of the data that allowed us to test kind of the cross-population generalizability of this fine-tuning strategy as well. So the fine-tuning strategy, we tried like several different things, a few different options. One of them is to throw kind of each individual with their paired genetic data and transcriptome data for a given gene, just kind of throw them into the training. So each one is kind of a separate sample. Each person is a separate sample for each gene. And we also tried kind of a more contrastive pairwise training approach where the loss function is based on comparing the difference in expression between two individuals. And so there we would get predictions for two individuals, and then we would train either on the actual delta between the expression levels of the two people from the measured data, or we would do a classification problem where we asked the model to say which of the two individuals has higher versus lower expression. So we tried kind of those two different pairwise training approaches. Okay, and then as I mentioned, for evaluating if this type of strategy improves performance, we held out different sets of individuals. So we first, for some of the genes, we held out kind of random individuals from the full Ioannidis data set. That's telling us kind of how well this fine-tuning approach generalizes to, just randomly held out individuals in the future. We also we did some genes where we held out all the Yoruban individuals. So they only saw variation from the European individuals. And this is motivated by some of these TWAS approaches that I had mentioned, like the PredictScan model and other similar TWAS models, where it's been observed that the transferability across populations is not as good as within a population. And partly due to kind of the LD structure being different with different populations, that if you train a variant-based model to predict expression in people from one ancestry, it has lower performance when you hold out people from kind of another population or another ancestry, probably because you don't have kind of all of the causal variants in the model correctly. So we have this Yoruban split where we're kind of evaluating whether we see that also when we do this fine-tuning with the deep learning models. Okay, and then we have a bunch of genes also that the models just never saw during fine-tuning, and so they have not seen any variation around those genes, where we held out kind of all the individuals for that gene during this fine-tuning process. Okay, and so the results of doing this, I will first just kind of orient with the two kind of baselines that we have. So on the left is the original informer model before any fine-tuning, and the performance on these kind of the random split, population split, and then the unseen genes. And so they're all kind of within error of each other. And then on the right-hand side is the performance of the PredictScan style linear variant-based model. And there you see the performance on the random split genes. So this is on randomly held out individuals randomly held out individuals for these PredictScan style trained models. So they've seen variation from the same gene, and they've trained these betas for the individual variants that are seen around each of the genes. In blue, either trained on random set of individuals, and then in orange is the population split where they've been trained on just the individuals from the European populations and then evaluated on the Yoruban population. And so this decrease in performance reflects what has been seen before with these TWAS style models. And it's kind of what we expect for the drop-off for this cross-population transferability. And then this style of model, you can't apply to unseen genes at all, because the way it works is by actually fitting a beta for a given variant from seen data for that variant. So it can't be applied to unseen genes. But that's kind of where we hope that the deep learning models should be able to improve performance there. Okay, so here I'm showing kind of 3 of the different fine-tuning strategies that we had and the performance of those. And so if you look at performance there, actually it is pretty similar for the different strategies, and it also looks very similar to what we had on the right for the just the variant-based linear model baseline. So having seen genetic variation kind of during this fine-tuning process, it does kind of improve the performance and the cross-individual correlation for the informer model in this case across individuals when you hold out individuals from the same genes. So for the blue and the orange case, with the randomly held out individuals and the population split individuals. So you do improve performance over kind of the baseline informer there, but what you get is something similar to what you see from this kind of linear variant-based baseline. And so it seems like by doing this fine-tuning, the models have been able to learn the effects of the variants that they saw during the fine-tuning process, and especially the ones that were kind of common and similar to what you would see if you just explicitly trained a beta for those variants. But they're not really exceeding the performance of the variant-based baseline at the moment with this strategy. And similarly for the population split, there's a similar drop in performance. There's maybe a very slight improvement over the baseline variant-based model in a couple of the fine-tuning strategies, but it's pretty small improvement. To get an improvement here, that can be due to doing a better job at identifying causal variants, because when you have a bunch of variants in LD with each other, this kind of variant-based approach, the TWAS approach, doesn't really have much basis for choosing one or the other of the variants in LD to include in the model. Whereas if the informer model or the sequence-based kind of deep learning model is doing a better job, if it has some prior about regulatory grammar, that's enabling it to do a better job of kind of selecting the causal variant among the set of variants in LD, then you would expect it to do kind of a bit better on this population split task. But we don't see a huge improvement there, kind of maybe only this marginal improvement, suggesting maybe there is some hint that the having this prior is enabling you to pick the better causal variant, but not a huge impact. And then for the unseen genes case, we unfortunately didn't see really any improvement over the kind of baseline informer model before any fine tuning. And so our kind of conclusion from this fine tuning process and this set of analyses is that if you fine tune the models using this kind of population variation that's available across individuals, the models end up learning the effects of the variation that's seen kind of multiple times across these individuals. But because there's enough shared variation, it makes it almost kind of too easy for the models to just learn the specific variation there. And it has unfortunately not kind of changed the model parameters in a way that the embedding is now more generalizable to new variants that are seen in other genes. And so this strategy of training across individuals on its own is not solving this unseen gene problem. And we think kind of additional strategies are needed. Maybe subsetting to rare variants with large effects, for example, could be a way to go rather than allowing the model to kind of just pick up these common effects that are seen multiple times in the training data. Okay, so we're kind of still exploring kind of options for how to incorporate variation kind of during model training. But something we were looking at in parallel, kind of motivated by this observation of the issue with assigning effects to kind of distal variants that weren't very close to the gene start site. Based on kind of that observation and what has been shown about these models and distal effects in general, we decided we should look at other phenotypes as well for this kind of personal variation problem. And so everything I've shown so far has been with gene expression, which is a phenotype that's pretty challenging and has kind of distal regulatory components to it. What if we look at something like chromatin accessibility that's a bit more proximally regulated? And so here we just did the kind of same setup that we had before, where we're going to have paired kind of personal genetic data and accessibility data. And in this case, we used a data set that was provided by Stephen Montgomery's lab at Stanford, where they had done attack sequencing in, again, these are lymphoblastoid cell lines, also from 1,000 genomes populations. These are from 5 different African populations that they did this sequencing. And so we have, it's a relatively small set of individuals, but enough to do this analysis. So we have these, paired personal genome and accessibility measurements. And they had also run already a QTL analysis with the attack-seq data. So we have a set of peaks where ataxic peaks where we know there is some genetic association with the height of that peak across individuals. And so we kind of subset the analysis, similar to what we did with the expression case. We subset the analysis to these ataxic peaks where we know there's some kind of genetic regulation going on. Okay, and so now we want to just set up the kind of same problem or same analysis that we had in the expression case, where we have this kind of individual by peak matrix of all the predicted accessibility. And then we have the kind of corresponding matrix of measured accessibility individuals by peaks as well. And so if we look now at the correlation across individuals for each of the peaks in this data set, we get now something that looks much more promising than we had for the expression case. So remember, for the expression, the kind of mean of this histogram was very close to 0. This is the histogram across peaks now of all of these cross-individual correlations. Okay, and we see in this case, this is again for Informer, the mean kind of cross-individual correlation for accessibility is around 0.3, so significantly non-zero and positive, which is great. And if you look at the distribution, it does kind of look like there's maybe this subset of peaks where you do have something that looks kind of similar to that expression case with this very wide distribution of correlations, including some strong negative ones, which I'll get back to in a second. But the kind of overall performance is looking pretty strong in terms of the models being able to handle this cross-individual task for this other phenotype, accessibility, rather than expression, presumably because of the more local regulation, but we're kind of exploring the details of that. So this is one example for Informer. We performed this analysis for several different models and also for different context sizes. Because it's accessibility, we don't necessarily need the full input context for each model. And so we looked at this mean, the mean of this distribution of cross-individual correlations for different models and different context sizes. And so that's what plotting here on the y-axis is this mean. And these are the kind of values that we get as a function of context size for a few different models. So just to point out, I guess, a couple of things. This is the Informer predictions in the orange or yellow color. The predict scan, so we again, even though predict scan and those kind of models were designed for expression, you can just apply the same framework with accessibility as the quantitative trait that you're predicting. And so we trained our own kind of linear variant-based model baseline using the same kind of setup as predict scan for accessibility. And we considered SNPs within different windows or context size around the peak. And so that's this kind of blue baseline here, which previously was for expression, that was the mean correlation was much higher than for the sequence-based models, which were all close to 0. Here, they're all much more tightly, the performance is a lot more similar between all of the models that we tested. And it seems to plateau after context sizes around 10 or 20-ish kb, which is consistent with not needing too much distal information to make these predictions. And I should note that we tried alpha genome as well for a couple of different context sizes. And we do see that for this latest model, the mean performance is a little bit better now than the variant-based baseline. So it is kind of slightly beating this out. And that's despite having this subset of peaks where there's this negative correlation. So if you remember from the kind of Informer distribution here, this set of negatively correlated peaks is contributing to the average, of course. And that set of peaks does exist still for the alpha genome predictions as well. And so they're kind of bringing down this overall average a bit, but it's still slightly better than predict scan. And to kind of look in more detail on kind of what's going on for different types of peaks, we did a similar analysis with the pairs of models that we had done in the gene expression case. So this is looking at if you have kind of for a given peak, accessibility peak in the genome that we analyzed, the cross-individual correlation from one model versus another. Okay, and so first showing that for alpha genome predictions versus predict-scan, so the kind of linear baseline predictions. And if you look at the scatter plot here, remember previously we had these X plots for the different pairs of models. If you have predict-scan, which is trained directly on the variants from a given gene, it tends, it doesn't have significant negative correlations generally. So it's more of a V plot in this case. But you can see that alpha genome does still have kind of this set of peaks for which the direction is predicted incorrectly. But within the kind of upper right quadrant where the direction is predicted correctly, there's a shift towards a bit higher performance. So there's kind of a shift away from y equals x to the right, which are peaks that alpha genome is doing better than this linear model baseline at explaining variation. So presumably picking up additional contributions from rare variants in those cases. If we look at alpha genome versus informer, so two sequence-based models, we actually do see this kind of X-like background again. So the majority of the peaks, since we do see that most of the peaks are well predicted for accessibility, most are in this upper right quadrant where both of the models, informer and alpha genome, are getting them correctly. But this kind of background set that we had seen in the histogram of peaks that do have these kind of strong negative correlations, it seems like those peaks have some characteristics that are similar to what we saw in the expression case, where if you compare the two models, the informer and alpha genome, you do kind of see this X-type shape for that set of peaks. So for those peaks, there's something going on that's very similar to the expression case where the models are having trouble kind of assigning direction of effect and getting it somewhat arbitrarily assigned between different models. So we're currently investigating kind of what's going on with that set of peaks and what are kind of the characteristics that explain what's happening in that case. But because you have this strong performance in general for accessibility, we think kind of a way to go forward for predicting personal expression variation and explaining gene expression across different individuals is to have kind of a hierarchical modeling approach where you first use these sequence-based models to predict local phenotypes such as accessibility. And then you can have kind of a second stage of a model where you use some known enhancer gene lengths, for example, and kind of put those more local predictions together to predict something like expression. So that's kind of a strategy that we're hoping to pursue. Okay, last thing, maybe in the last 5 minutes, I will just quickly mention some additional work that we've been doing recently and hoping to have a preprint soon, which would be great to get people's thoughts on. So we have been thinking about in general kind of what other type of information is available for personal genomes that we could use to kind of augment the standard sequence to function type approach. And through some collaborations, we've been looking at kind of long read sequencing data, which is becoming more common. And as you start having long read data for patients and in the clinic, you're going to, along with the sequence, just get information on the CpG methylation. It just kind of comes along with the data that you get from kind of reading out the information and the kind of the way the long read data is generated. You have CpG methylation status along with sequence. So we've been working with a couple of students in the lab and jointly advised with Aaron Streets. So this is all kind of a collaborative project with Aaron's group, where we're augmenting the existing sequence input models with the additional CpG methylation information as well, and seeing kind of where that improves things and where it doesn't improve things. And it's kind of all related to this somewhat interesting question of the sequence only models, kind of before you add in methylation, how much have they already learned about CpG methylation? And some of it is inferable from sequence, because if the CpG methylation was established kind of early on in development in different cell types, let's say, if it was established due to some sequence patterns and sequence motifs that recruited various machinery to put the methylation there, then some of it may be learnable from sequence, and the models have already learned some of this information, but not all of it. And so that's what we've kind of found to be the case in some of the experiments that we've run, is that some of this methylation has already been learned and some not by the sequence-only models. And so this kind of architecture that we have come up with for kind of incorporating methylation and sequence into the same modeling framework is to use kind of any pre-trained sequence model. So we don't want to have to retrain all of the or recreate, a good sequence embedding when we already have all the existing Informer Borzoi alpha genome style models that generate nice sequence embeddings that have already been trained with lots of molecular data. So we have kind of an architecture where you can kind of substitute in any pre-trained sequence encoder to get an embedding of the sequence context or the sequence information. And then we kind of split that embedding into two different portions, one of which gets conditioned by the methylation state of that same sequence. So we have kind of the paired methylation information from the sequence, and we use it as a kind of conditioning vector to element-wise multiply part of the embedding space from the original sequence. And so we've kind of split this into two because of the some of the sequence and some of the information already captured from sequence is related to methylation and some is not. And so we found that this kind of split approach works pretty well. So what we can do here is, say for a given sequence, we can either use the kind of the methylation that was observed from a particular person's genome from the long read data, or you can plug in kind of an imputed CpG methylation, or you can do, for model interpretability and some kind of synthetic experiments, you can plug in different methylation landscapes and see kind of what the effect on the predictions would be from different methylation patterns. And so kind of overall, we see that the model conditioned in this way with the methylation information is improving predictions of accessibility, particularly if you look at kind of the difference between different cell types, which makes sense that some of the difference between cell types has been encoded into this kind of epigenetic pattern of methylation. And if you can capture that in the model, you can kind of improve the ability to understand differences in accessibility between different cell types. And you can also use this to predict differences between different haplotypes for cases where there's different methylation on the two different copies of the chromosome, which you can't really do with the sequence-only model. Okay, since I'm running out of time, I'll just quickly wrap up by saying, when we look at kind of where in the genome, so which parts of the genome are contributing to these kind of improvements in performance from this type of methylation conditioned model, we see a lot of the regions of the genome are performed similarly. This is kind of comparing performance from unconditioned and conditioned model on a cross cell type prediction task. So there's a lot along the diagonal that aren't improved, but then there's kind of this subset of regions of the genome where the methylation information is adding something. And so that's what we're kind of digging into in terms of what is the kind of biological interpretation of what's going on there. And again, these are regions not necessarily where methylation, or these are not only the only set of regions where methylation is kind of causally affecting accessibility, but it's places where the sequence-only model has not captured the effect of the methylation. So it's maybe methylation that was established from kind of longer distance propagation of methylation early on. And so there aren't kind of clear sequence patterns that the sequence-only models have learned to explain it. Okay, this is kind of an example region where we see target kind of accessibility prediction, or this is accessibility measurements, and then the predicted accessibility in blue here. And you can see kind of a difference in how well the conditioned versus the unconditioned models are able to kind of recapitulate the true accessibility in that region. And part of it is due to this kind of difference between what the true methylation landscape looks like and what the sequence-only model was able to learn about the methylation landscape. And because it's kind of different in these regions over here, that explains why the kind of sequence-only model is not doing as well as predicting the true accessibility in those regions. Okay, so I will wrap up and just say that we're kind of working on applying this type of approach to some data from rare disease patients as well. And sometimes there are there are methylation changes in kind of rare disease patients that are contributing to their disease phenotypes. And you can improve the kind of diagnosis of those kinds of cases with this type of model. So we're working with some collaborators to do that. Okay, so I will wrap up there, and I want to acknowledge kind of everyone in the lab who's worked on kind of all these projects, and then also mention that there are several other groups that are doing kind of very similar work and asking similar questions. The original kind of work on the personal expression prediction we kind of published jointly with Sarah Mostafavi's group, and we've also been talking to kind of Katie Poller, Alexis Battle. They've been thinking about these problems for a long time as well, and so we've had great conversations with them and people in their groups. And the kind of recent accessibility work is joint with Anshul as well, and then Aaron for the methylation project. So thank you very much, and happy to take questions and chat more about this.